No, actually, we were doing antibiotics today. Pardon me. The introduce. Antibiotics. How does the antibiotics work? First of all, one way they work is to prevent the bacteria from making normal cell walls. In the bacterial cell walls, water constantly enters by osmosis. So how does the water in active transport, diffusion, active transport, transport يعني osmosis. Osmosis is the one responsible for water, and right, so the water constantly enters by osmosis, which is from high water potential to lower water potential across the water potential, or down the water potential, sorry. This enters of water would normally cause the cell to burst, osmotic lysis. It doesn't burst because of the cell wall that surrounds all of the bacterial cells. This wall is made of tough material that isn't easily stretched. As water enters, the contents expand and push against the cell wall. Certain antibiotics kill bacteria by preventing them forming cell walls. They inhibit the synthesis and assembly of the important peptide cross-linkage in the bacterial cell walls. This weakens the walls, making them unable to withstand pressure. As a result, they are unable to prevent water entering, and osmosis lysis occurs. Put me in the comment how much I said cell walls in here. Let's count them. Cell walls, cells only, cell wall, cell wall, cell wall, cell wall, cell wall, wall, and I believe there is some that I didn't see. Can you see how important it is? I see. There are lots of cell walls everywhere. Why are these cell walls important? Because this is how we prevent, or how we burst, or not we, pardon. So what does it have to do? Let me first choose a good, beautiful color. You like this one? Put me down in the comments below. Bye. Antibiotics. How does the antibiotics occur? First of all, they prevent the bacteria from making normal cell walls. Why do they don't want it to make normal cell walls? Because in the bacterial cells, water constantly enters by meiosis, and this entry of water would normally cause the cell to burst, which is basically the osmotic lysis type. It doesn't burst because of the cell wall that surrounds all of the bacterial cells. The wall is made of tough material that isn't easily stretched. As water enters, the content expand and the push against the cell wall. Why do we want to prevent bacteria from making normal cell wall? Because certain antibiotics kill bacteria by preventing them forming cell walls. Why? They inhibit the synthesis and assembly of the important peptide cross-linkage in the bacterial cell walls. This weakens the walls, making them unable to withstand pressure, and as a result, they are unable to prevent water entering, and osmotic lysis occur. That's great. So that's why. Read that only highlighted part. How antibiotic works. They work by preventing bacteria from making normal cell walls. Why is that? Because they inhibit the synthesis and assembly of the important peptide cross-linkage in the bacterial cells, and so this weakens the walls, making them unable to withstand pressure. As a result, they are unable to prevent water entering, and osmotic lysis occurs. If you don't understand, do not worry. We're doing it once again. No, not this, sorry. This. How antibiotics work. They work by preventing bacteria from making normal cells. But why is bacteria making normal cell walls something dangerous? Because the water enters by osmosis, and the entry of water would normally cause the cell to burst, which is basically the osmotic lysis. It doesn't burst because of the cell wall that surrounds all of the bacterial cell. The wall is made of tough material that isn't easily stretched, and as the water enters, the content expand and push against the cell wall. So if they don't have a cell wall, the water won't expand and push against the cell wall. Certain antibiotics kill bacteria by preventing them forming cell walls. They inhibit the synthesis and assembly of the important peptide cross-linkage in the bacterial cell. What do? They inhibit the synthesis and assembly of the important peptide cross-linkage in the bacterial cell wall. What does it do? It weakens the cell wall, making them unable to withstand pressure, and as a result, they are unable to prevent water entering. The osmotic cell lysis occur. Okay. What about the antibiotic resistance? We're going to also talk about it here, antibiotic use and resistance. So it's not only this part, do not worry, Yani. Antibiotic resistance. Antibiotic resistance. Shortly after discovery, it became clear that the effectiveness of some antibiotics at killing bacteria was reduced. Shortly after discovery, it became clear that the effectiveness of some at killing bacteria was reduced. So antibiotic bacteria was reduced after the discovery. So why is the effect of the antibiotic decreasing? The populations of bacteria had developed resistance. That's the reason. To antibiotics such as penicillin due to chance of mutation. For example, we have this bacteria which has this DNA gene, whatever, and there is this antibiotic which is made for it, and then this DNA got mutated and became black. So it's no more like this one, it became resistant to it, right? Or whatever, either way. In the case of resistance to penicillin, the mutation resulted in certain bacteria being able to make the new protein which broke down the antibiotic penicillin. This is how, for example, penicillin was transmutated or whatever and what was resistant. The gene for penicillinase and hence antibiotic resistance is passed from one generation to the next by what we call the vertical gene transmission. The allele for the resistance is carried out on the circular loops of DNA called plasmids. These can be passed from cell to cell by conjugation. Resistance can find its way into another bacterial species by horizontal gene transmission. Horizontal gene transmission can lead to certain bacterial accumulation DNA that gives the resistance. Do you understand? Do you know how important these paragraphs are? They're important information so far. Like, shortly after discovery, it became clear that the effectiveness of some antibiotics at killing bacteria was reduced. First thing, that their effectiveness of some antibiotics at killing bacteria was reduced. There's a chance that antibiotic effectiveness gets reduced. How did they know that? The population of bacteria had developed resistance to antibiotics such as penicillin due to chance of mutation. How was the penicillin chance mutated? In the case of resistance to penicillin, the mutation resulted in certain bacteria being able to make new protein which broke the antibiotic penicillin. The gene for penicillin case and hence antibiotic resistance is passed from one generation to the next by vertical gene transmission. From one generation to the next, vertical gene transmission. Resistance can find its way into other bacterial species by horizontal gene. So one generation to the next, same species, vertical. Different species, horizontal. The allele for the resistance is carried out by circular loops in the of DNA called plasmids. So the allele of the resistance is carried in circular loops of the DNA called plasmids. These can be passed from cell to cell by conjugation. Horizontal gene transmissions can lead to certain bacterial accumulation in DNA that gives them resistance to a range of antibiotics. Let's read each color alone because it's important. First of all, effectiveness of some antibiotics at killing bacteria was reduced. Population of bacteria had developed resistance to antibiotics such as penicillin due to chance of mutation. This happens as the bacteria being able to make new protein which broke down the antibiotic penicillin. Basically, the allele for the resistance carried out on circular loops of DNA called plasmids, and these can be passed from cell to cell by conjugation. Resistance can find its way into other species by horizontal gene transmission. Being passed by conjugation can also happen in the vertical gene, which is basically from one generation to another. In the not conjugation, the horizontal gene, vertical gene, and horizontal gene transmission can lead to certain bacterial accumulation that gives them resistance to change of antibiotics. Last thing before we finish this chapter is basically the antibiotics use and the resistance. We've talked about the antibiotic resistance in general, but there is also something called antibiotics resistance and tuberculosis and antibiotics and the MRSA. The antibiotic resistance and the tuberculosis, basically one problem with the antibiotic treatment for the TB is the long period for which the antibiotic must be taken. Again, one problem with the antibiotic treatment for the TB is the long period for which the antibiotic must be taken. It takes long, for example, months or whatever. After a number of months, the patient feels better because of the majority of mycobacterium that has been killed. they then feel tempted to stop taking the antibiotics. This is a problem as the few bacteria that remain are those most resistant to the antibiotic. There is a selection pressure that leads to the development of strains of mycobacterium that don't respond to the antibiotic. These strains intercharge genes for resistance by conjugation. To overcome this problem, a cocktail of 3 or 4 antibiotics is used to ensure at least one will be effective. Then we have the antibiotic resistance and the MRSA. Many people carry a bacterium belonging to the genus Staphylococcus in their throats. Latin stuff. Anyways, MRSA is the name given to any strain of this bacterium resistant to one or more antibiotics. So what's the name given? MRSA. It's especially prevalent in hospitals and can present danger because people in a hospital are older, sicker, and weaker, making them more vulnerable. Many people live close together, which is perfect for transmission. Many antibiotics are used in hospitals, and so any mutant-resistant strain has an advantage over non-mutant strains. And there are some implications of antibiotic uses. The antibiotic resistance is on the increase for a number of reasons. From antibiotics are used to treat minors, used to treat minors ailments whose symptoms are short-lived, to that they are sometimes used to treat viral diseases that may help prevent the development of secondary bacterial infections. Also, patients do not always complete the course of antibiotics, and also patients also stockpile unused antibiotics. Antibiotics are used in the treatment of minor ailments in animals, and they are used to prevent disease among intensively reared animals. Lastly, doctors accept patients' demand for treatments. This is the end of this video. In the next video, as I told you, we'll be doing the things we didn't cover in this video. Have a nice day.